Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, leveraging human primary cell in vitro assays to advance immune-targeted cancer therapeutics. My name is Dora Wells, and I'll be moderating today's presentation. So I'm delighted to be joined by Dr. Gavin Meehan. Dr. Meehan has a PhD in neuroimmunology from the University of Glasgow. Following his PhD, he spent almost a decade undertaking academic research, focusing on a range of topics, including scleroderma, malaria, rheumatoid arthritis, as well as SARS-CoV-2 and other related viruses. Currently, as a senior study manager at Antibody Analytics, he provides scientific advice for the design of customer studies and oversees a variety of projects. He has a particular interest in autoimmunity, T-cell biology, dendritic cells, and other antigen-presenting cells. After the presentations, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your question at the left of your screen. Just click on the purple tab. And now, without further delay, I would like to hand it over to Gavin for today's presentation, and I'd like to thank him for presenting to us today. Please go ahead, Gavin.
So thank you very much for the introduction. As discussed, I'm Dr. Gavin Meehan. I'm a Senior Study Manager at Antibody Analytics, and today I'm going to be discussing leveraging human primary cell in vitro assays to advance immune-targeted cancer therapeutics. Before I start, I wanted to give a little bit of background about our company. Antibody Analytics is a collaborative immunology CRO based in Scotland in the UK. We were founded in 2015 and we are located approximately 20 miles outside of Glasgow in a town called Motherwell. We've a recently expanded 24,000 square foot facility which houses more than 100 employees. In terms of expertise, we're a world leader in human immunology research service provision for the assessment of immuno-oncology, autoimmunity and biosimilar therapeutics. We apply a unique combination of immunology, bioanalytical and molecular biology expertise to establish advanced experiment solutions to assess the safety and efficacy of therapeutics. We not only specialise in antibody-based drugs, but also cell-based therapies and advanced medicines. And we support a global customer base who operate from the earliest phases of drug discovery to the clinic. Now, before we go into the specifics, I'd like to discuss what cancer is and why immuno-oncology matters. As I'm sure most of you are aware, cancer is a heterogeneous group of diseases characterised by unregulated cell growth. With over 200 distinct cancers affecting different tissues and organs, statistics have shown that between 1 in 2 and 1 in 3 of us will develop cancer within our lifetime, and it's therefore unsurprising that cancer remains the leading cause of death worldwide, with approximately 10 million deaths recorded in 2023 alone. There are various treatments for cancers, including surgery, radiotherapy and chemotherapy, but today I want to discuss immuno-oncology. Now, immuno-oncology, which is also known as IO, uses immune-based therapies such as antibodies or cell-based technologies alongside the body's own immune system to prevent and treat cancer. IO has taken off massively in the past 20 years and is diversified away from the original therapies like checkpoint inhibitors to now encompass cell-based therapies such as CAR-Ts, immunomodulators, as well as cancer vaccines. The increase in IO therapies is apparent in the drug development pipeline, with thousands of treatments entering the preclinical phase of development and significant increases in the number of IO-specific clinical trials year on year. I'd now like to touch upon what cancer is in relation to tumours. Tumours are not simply a group of cancer cells, but rather a heterogeneous collection of infiltrating and resident host cells, secreted factors, and extracellular matrix. Tumour cells stimulate significant molecular, metabolic, cellular, and physical changes to support tumour growth and progression. The composition of the tumour microenvironment can vary between tumour types, but hallmark features include stromal cells, blood vessels, as well as extracellular matrix. It also includes immune cells. Now, immune cells are a critical component of the tumour microenvironment, but a dichotomy exists in the behaviour of the immune cells, which can either promote tumour growth or can suppress it. Tumour microenvironment takes advantage of naturally suppressive immune cells such as Tregs and M2 macrophages and uses them to inhibit the immunostimulatory response, but it's also able to induce dysfunction of immunostimulatory cells such as T cells, which it can exhaust and cause them to become unresponsive. All these cells can be assessed using in vitro assays, but before I do that, I want to discuss why we use in vitro assays in the first place. In scientific research, there are three broad categories for performing experiments: in vitro, in vivo, and in silico. All three of these systems have their benefits and drawbacks, and collectively they're able to provide us with an overall picture of how a therapeutic can function. In vivo assays allow us to examine the effect of therapeutics in a whole physiological system, providing us with a spatial and temporal dynamic that would otherwise be hard to measure with the other systems. In silico systems consists of computer programs or softwares that allow us to model complex processes and process large data sets. In recent years, the rise of AI is giving us new methods to understand more and more complex questions. But of course, today we're here to talk about in vitro systems. So what are their benefits? Well, first off is the mechanism of action. In vitro systems allow the effect of a therapeutic to be determined against individual cell types. There's customization, where multiple variables can be assessed readily, including cell types, concentrations, and exposure times. This allows for high throughput analysis, where large numbers of samples can be assessed quickly and efficiently. There's also ethical considerations. Using in vitro assays removes the need for work on animals. And most importantly, there's translatability. Therapeutics intended for human use can be assessed against human cells in vitro assays. This provides much more robust information than either of the other two categories can allow. So what are the important factors that should be considered when you're trying to assess therapeutics using in vitro systems? Well, first, we need to know what the target of the therapeutic is and whether it's expressed on one or multiple immune cells. This can affect your ability to interpret data. It's also important to understand the mechanism of action and how changes in this mechanism can be measured. And ultimately, it's important to have an idea of how your therapeutic is likely to impact cellular function. Now, with all of these in mind, here are some basics for good assay design in immuno-oncology. First, we need to isolate or generate the cells of interest. This may seem obvious, but some cell populations are very small and it may be difficult to generate sufficient numbers. Secondly, we should phenotype the cells. This will demonstrate that the cells have been generated correctly. At this stage, it's also important to examine the expression levels of the receptor. This is incredibly important and something I'd really like to stress. Even when receptor levels are theoretically known from the literature, it's incredibly important that you establish the levels of receptors in your system. This ensures that if your new therapy doesn't work, it isn't due to the absence of the receptor. Lastly, it's important to assess the impact of your therapeutic on cellular function. This can be direct or indirect. By this, I mean that you can look at the impact of the therapy on your target cell itself, or the impact of another cell that your target cell influences. This could be activation or suppression mechanism, depending upon your cell type. And we can also look at the impact of a therapeutic on a cellular specific mechanism. This could be phagocytosis in macrophages or dendritic cells, antibody production in B cells, or mitosis with neutrophils. The important part is that therapeutics can be added at any stage of this process and can be assessed for their abilities to change the outcomes. Before discussing specific assays, I just want to show you the type of assays we're able to look at within our company. With three main departments, there are many aspects of drug development a company like ours is able to assess, but today we're going to focus on three specific areas within the context of immuno-oncology. In immune cell biology, we'll examine screening, as well as understanding mechanisms of action, and in cell line development, we'll examine target cell lines. With this in mind, I'd like to discuss immuno-oncology in vitro assays. Now, we're able to examine any of the individual immune cells shown here using our standard assays. However, I do think it's worth mentioning that almost 80% of our work in the immunology department is bespoke, with assays designed and tailored to the needs of a customer. This flexibility is really vital for trying to better understand the impact of a therapeutic and trying to better evaluate its effects. Now, as much as I'd love to go through all the different assays, for the sake of time, we're going to focus on some specifics. These include assays on macrophages, Tregs. We're then going to change tact and talk about the importance of cancer cells and target selection. And lastly, I want to discuss our work focusing on the tumour microenvironment as a whole. So let's start with macrophages. It's first important to know how macrophages behave in the tumour microenvironment. There are two broad categories known as M1 and M2. M1 macrophages are inflammatory and tumour suppressive. They're able to kill and phagocytose cancer cells, which leads to antigen presentation to other immune cells, including T cells, which results in a larger immune response. In contrast, M2 macrophages are immunosuppressive and tumour promoting. They suppress the inflammatory response of other cells and secrete growth factors, which leads to tumour growth. In cancer, M2 macrophages are found in abundance in the tumour microenvironment, where they're known as tumour-associated macrophages, and it's therefore no surprise that TAMs are a major focus of new immuno-oncology therapeutics. Based upon our assay design basics we discussed earlier, we first have to generate our macrophages. We have access to a number of on-site cryopreserved PBMCs, which we can use to generate monocyte-derived macrophages. We do this by isolating CD4 monocytes using magnetic isolation, which we then culture with MCSF. This generates resting state or M0 macrophages. These can be further polarized with different cocktails to produce specific subtypes, including M1, M2A, M2C, and M2D, depending upon the specific disease state being studied. Now that we've generated our macrophages, we need to phenotype them. We do this using flow cytometry, which can demonstrate differences between the various subtypes. From these histograms, for example, you can see that HLA-DR and the co-stimulatory molecules CD80 and CD86 are more highly expressed in the M1 subtypes, which is in keeping with their pro-inflammatory state. In contrast, the M2C macrophages express high levels of the scavenger receptor CD163, which is associated with anti-inflammatory processes, which again is in keeping with the state of this subtype. We can also assess the macrophages through their cytokine secretion using ELISA. In this example, you can see that M1 macrophages produce high levels of TNF-alpha compared to the M2 subtypes. And test articles can be assessed at any of these polarization stages to determine whether they can influence polarization to a particular state. Alternatively, we can add the test samples to macrophages that are already in a different state to see if we can switch them into a more pro-inflammatory state like M1s. This is all due to the plastic nature of macrophages and is of great interest to a number of drug manufacturers, as macrophage reprogramming offers a real potential for treating cancers. So now that we've generated and phenotyped our cells, we need to assess their function. With macrophages, we're able to do this using a host of T-cell activation assays. The example I'm going to show here is a mixed lymphocyte reaction, also known as an MLR. The polarized macrophage subtypes are mixed with pan T cells from an allogenic donor in the presence of test articles. The difference in the HLA status between these donors drives T cell activation. Again, test articles can be included either in a pre-stage or within this assay itself. And we can assess the T cell responses, so we can look at T cell proliferation, or we can use ELISA or multiplex cytokine analysis to examine the cytokine production. In this example, you can see that by using Ki67, we're able to assess proliferation of the CD4 and the CD8 subtypes. M1 macrophages lead to significant proliferation in both of these populations. It also leads to significant increase in the production of interferon gamma in these cells, whereas the M2 anti-inflammatory macrophages produce much lower levels. This system is particularly well suited to assessing macrophage reprogramming, although it could be used to assess other therapeutic strategies. T-cell suppression is another readout that we can use for assessing macrophage polarization. In this assay, PBMCs are labelled with a proliferation dye and activated with PHA. PBMCs are mixed with macrophages in a 1-to-1 ratio, and the impact upon PBMC proliferation is assessed by flow cytometry. As you can see, the M2C macrophages are particularly suppressive compared to the M1s and the other M2 subtypes. And they are very similar to the T-cell unstimulated population. When compared directly to the M1 macrophages, they're also shown to be suppressive more than the other M2 subtypes. Again, test articles can be included within suppression assays to test the ability to induce or reduce suppressive capacity across the different subtypes. As I discussed earlier, there is also cell-specific mechanisms can be assessed. We have multiple assays to assess phagocytosis, including using labeled beads and FABZAP assays. But I'm going to discuss antibody-dependent cellular phagocytosis here, also known as ATCP. In this assay, the effector cells are any of our macrophage subtypes, and our target cells are CD20 expressing cell line. Here we've used Raju cells. The test articles is rituximab, which is an anti-CD20 therapeutic. And the Raju cells are labelled with a cell tracker dye, whilst the macrophages will be labelled with a surface marker antibody during the flow cytometry preparation. Flow cytometry is used to assess the double positive population, which acts as an indicator of ADCP. As you can see here, rituximab induces a dose-dependent increase in ADCP with increasing concentrations, with M1 and M2C macrophages being the most phagocytic, whereas the M2 macrophages have the lowest levels. In the system, target cells can be substituted for any relevant target-expressing cell line, and these can be immortalized cell lines, or we could generate a cell line for you depending upon your target. So the endomacrophage section, I just want to discuss the different kind of therapeutics that can be assessed using these assays. These include therapies such as Siglec-10 inhibitors, which can restore the anti-tumour function of TAMs. These could be assessed in an activation assay. We could look at mechanisms such as anti-PD-L1 antibodies, which can limit the tumour-promoting activity of TAMs. We can use assays to assess changes in TAM recruitment with a number of chemokine inhibitors. These would use transwell assays, which we also have established in the company. We can look at the ability of certain therapeuticals like CAR-Ts for the depletion of TAMs. And as we have already discussed, we could look at macrophage reprogramming using the assays we've described. So now that we've finished discussing macrophages, I'd like to move on to Tregs. Regulatory T cells, known as Tregs, are a CD4 T cell subset that are characterized as having constitutive expression of the transcription factor FOXP3 alongside high levels of CD25. Tregs are essential in maintaining immune homeostasis and self-tolerance. This function is fundamentally important to normal health, with animal models that lack Tregs developing severe autoimmune disease and experiencing premature death. There are two types of natural Tregs, thymus-derived Tregs and peripherally induced Tregs. In cancer, the suppressive nature of Tregs is a major contributor to tumour growth, so it's therefore no surprise that they are a major target of new immuno-oncology therapeutics. Tregs are a little bit different than other cell types as we have two different methods for producing them, isolation and generation. N-Tregs can be isolated directly from PBMCs using magnetic isolation. As they only comprise 2% of the PBMC population, though, they do require a 21-day expansion before there are sufficient cell numbers for the assays. Alternatively, we can generate induced Tregs by first isolating naive CD4 T cells from PBMCs. These can be mixed with an induction cocktail, and over 8 days, they'll produce sufficient numbers of Tregs. In terms of phenotyping, it's relatively simple. With N-Tregs, you can see that the FOXP3 and CD25 expression is checked in the initial isolation. Following the expansion, this population is in the same place, but is larger due to the increased cell number. With iTregs, we first isolate the naive T cells, which are CCR7 and CD45RA double positive. Following our iTreg induction process with our cocktails, we can induce Treg expression markers such as FOXP3 and CD25, and we have a yield of approximately 85%. To assess the function of Tregs, we look at a T cell suppression assay. In these assays, Tregs are co-cultured with cell tracker labeled PBMCs, which are activated with anti-CD3 antibodies. Flow cytometry is used to assess proliferation of the conventional CD4 and CD8 T cells. As with our other assays, test articles can be added at any stage, either during the Treg induction or within the assay itself. As you can see here, the iTregs significantly inhibits T cell proliferation with higher cell numbers inducing high levels of suppression in both the CD4 and the CD8 populations. And with the histograms, you can see that the 1-to-1 ratios are similar to the unstimulated conventional T cells. Wanted to show an example of these assays in action. This is a case study here that we performed in collaboration with Miltenyi Biotech. Here we focused on CBL-B, which is an E3 ubiquitin protein ligase that negatively regulates Treg activation. As with the previous assay, Tregs were labelled with cell tracker dye and anti-CD3 stimulated PBMCs in the presence of a CBL-B inhibitor. As you can see, the inhibitor reverses the Treg-mediated suppression with both the CD4 and the CD8 T cells, which can be seen in particular in the histogram with the high doses, which look similar to the no Treg controls. Again, I'd like to give examples of the types of therapeutics which could be assessed using these assays. Treg co-receptor blockade could be assessed, as could inhibitors to Treg signaling and suppression assays as shown in the previous slide. Negative immune regulators and inhibitors of Treg secretion and cytokines could also be assessed with suppression assays. We can look at Treg metabolism, And as with macrophages, we can also look at Treg migration using transwell assays. Now I'd like to change direction and discuss the cancer cells themselves. So one of the most important aspects of assay design is the selection of the target cancer cells. Basic assays select target cells that express the tumour-associated antigens. This is a perfectly acceptable approach, but it doesn't take into account certain aspects of cancer, and one of these is antigen density. One of the key challenges in developing tumour-targeting therapeutics lies in identifying optimal tumour-associated antigens. Often these target antigens are not cancer-specific. They are overexpressed in cancer cells, but can also be found in healthy tissues. This can lead to what are known as on-target off-tumour effects, which can lead to unwanted toxicity. This heterogeneous tumour antigen expression not only makes efficient treatment more difficult, but can also contribute to immune evasion by tumours that downregulate expression of the target antigen to evade destruction in a process known as antigen escape. This challenge applies to multiple immuno-oncology therapeutic modalities, including immune cell therapies, immune cell engagers, ADCs, and conventional antibodies. The expression of target antigens in normal tissues, as mentioned, can lead to off-tumour toxicity, which is a common side effect associated with many immune-engaging therapeutics. In particular, on-target off-tumour toxicity has become a significant concern, and regulatory guidelines have been established to assess the undesired targeting of healthy tissues that express the target antigen, as you can see here in the snapshot from the FDA guidelines for the development of CAR T cell therapeutics. The conventional approach to assess the impact of antigen density and the efficacy of therapeutics is the use of panels of cancer cell lines that express differential levels of the antigen of interest. These cells are employed in the functional assays, and it's typically expected that the observed response should be proportional to the number of receptors. This approach has various limitations, however. Firstly, the cost of setting up an appropriate panel of cancer cell lines can be prohibitive and can reach tens of thousands of dollars if licensing costs are not included. Secondly, the range of expression, even in large cell line panels, can be limited, which does not allow for the determination of the threshold of activation. And lastly, the cell line background can introduce variability, as intrinsic differences between cell lines can render them susceptible or resistant to the therapeutic in a manner independent to the antigen expression level. I'd like to draw your attention to this publication that effectively highlights the limitations of conventional approaches when assessing antigen density in the context of CAR T therapies. In this study, the authors aimed to fine-tune the effector function induced by HER2 and EGFR targeted CAR T cells by modulating the affinity of the antigen binding moiety of the receptors. They showed no correlation between receptor numbers and effectors when different cancer cell lines were used. However, when they induced differential expression levels on cells on the same background, they showed a correlation between the effect and the receptor number. So in order to address the limitations of the conventional approach to assessing antigen density, we needed a better system. This system should have the following characteristics. It should be a single cell background to ensure the absence of confounding factors from heterogeneous cell populations. It should have a large dynamic range of expression. This enables the assessment of antigen abundance across a wide range of expression levels, ranging from hundreds to hundreds of thousands of receptors per cell, which should provide a more comprehensive analysis. It should be finely titratable. The ability to finely control antigen expression levels is crucial for understanding the impact of varying densities on therapeutic efficacy. And there should be an availability of a negative control, which is essential to establish a baseline and validate your experimental outcomes. It should also be compatible with immune cell bioassays. The ideal system should be easily integrated into existing bioassays, otherwise the system will be of little use. So to answer all these points, we developed an inducible platform called Index2. In this platform, we have a finely titratable system for inducing antigen expression. We have associated functional assays, and used together, we can determine receptor thresholds, which is the minimum number of receptors required for a particular therapeutic to induce a response. So we developed the Index2 platform as a chemically induced proximity CIP system for inducible and titratable expression of one or two antigens in a controlled manner. CIP systems work by using small molecules to induce proximity of proteins, allowing dimerization of the protein partners. If one of the protein partners is fused to a DNA binding domain and the other to a transactivation domain, this creates a molecular switch that controls the expression of a transcript by modulating the concentration of the small molecule. Various CIP systems have been reported, such as the tacrolimus-based and juberolin-based systems that have been used in various applications. For the index 2 system, we chose the abscisic acid, or ABA, and indoloacetic acid, or IAA, plant hormone-based CIP systems due to their advantages, namely reduced background, high treatability, and the inert nature of the small molecules involved, which gives them an ideal use for cellular bioassays. We've made significant modifications to our chosen CIP systems to ensure efficient orthogonality, reduced background expression, and increased titratability. So I want to show you an example of the cell lines we generated. We usually employ the CHOK1 cell line as a background, as it's a non-human cell line and it will not present human antigens and functions somewhat as a blank canvas. Examples of the tumour-associated antigens we generated cell lines for include EpCAM, HER2, BCMA, as well as CD19, which, as you can see from the graphs, have a finely titratable expression. We also have a 2-antigen system. This is 2 antigens within a single cell line. CD22 is under the control of the ABA system, and CD19 is under the control of the IAA system. This is still undergoing refinement, but we're hoping to have some more data on this in the upcoming months. So I want to discuss a case study of an application of this system. Using a similar approach as the paper I showed earlier, we generated a high and low affinity CAR-Ts which targeted HER2. We have our HER2 cell line, of which we exposed these CAR-Ts to, and we measured cytolysis using the Excelligence real-time cell analyzer, and we measured interferon gamma using ELISA. Here you can see there's a receptor-dependent increase in cytolysis, as well as interferon gamma, particularly with the high affinity CAR-T compared to the low affinity one. We also have shown here what's called an R50. This is somewhat similar to an EC50, and it indicates the number of receptors that produce half the total cytolysis or cytokine production. This provides a method for comparing between the CAR-T affinities, which can be particularly useful. Is here you can see the high affinity has an R50 of about 560, whereas the R50 for the low affinity is in the region of hundreds of thousands. With interferon gamma, you can see the high affinity CAR-T was able to produce high cytokine production at high receptor numbers. However, the low affinity CAR-T produced little interferon gamma above baseline. The next case study I'd like to discuss is focusing on targeting of CD19 with a T-cell engager. As you're no doubt aware, there are several approved CD19 targeting therapeutics. Even though these therapeutics are very successful, a proportion of patients that are treated with them do not respond at all or relapse shortly after a period of time. One of the reasons that this happens has been linked to the expression of CD19 levels on the B cells in these patients. A paper by Spiegel and colleagues tried to correlate the levels of CD19 expression to the response of Yescarta treatment in large B-cell lymphoma. By employing quantitative flow cytometry, they defined that there is generally a higher chance of resistance when CD19 levels were below 3,000 receptors per cell. Beyond CAR-T, there's also been multiple papers looking at blinatumomab, which is also known as Blincyto, and B-cell acute lymphoblastic leukemia. So using these publications as a guide, we can define a broad area of expression levels that are linked to a lower or higher risk of resistance to therapy. So therefore, an ideal CD19 targeting therapeutic would retain efficacy at all levels of CD19 expression. This brings us to a therapy in development at Cullinan Therapeutics. CLN-978 is a T-cell engager targeting CD3 and CD19 that's been created for the treatment of B-cell malignancies. It contains an HSA binding domain to extend its half-life, and has been shown to induce high levels of cytotoxicity, T-cell activation, as well as the production of pro-inflammatory cytokines. This therapy was predicted to work in low expression levels of CD19, and therefore it has a use in relapsed B-cell malignancies. So we wanted to test this therapeutic within our index 2 system. So to do this, we took our CHOK1 cell line expressing CD19. We took T cells from 10 donors and set up an assay between the cell line and the T cells in the presence of Blincyto or CLN-978. Cytolysis was measured using the Excelligence real-time cell analyzer, and cytokine secretion was measured by ELISA. For this cohort of 10 healthy donors, we observed that CLN-978, which is shown here in red, induced higher levels of cytolysis at all levels of receptor expression and it was also able to induce interferon gamma production by the activated T cells, again at all levels of CD19 receptor expression. So in order to determine the receptor threshold of activation for the two molecules, we used an alternative analysis of the 10 donor data, known as receiver operating characteristic analysis. This was developed during World War II for radar signal detection, but has found applications in other fields, including diagnostic testing, where we can show the differences between positives and negatives. Here we use it to define a threshold which is considered true, and which we use as a threshold of receptor activation. Using the cytotoxicity into interferon gamma production as a readout, we compared CLN-978 and Blincyto, and it showed that CLN-978 for both readouts has a threshold of activation which corresponds to approximately 80 receptors. However, for Blincyto, the threshold of activation is quite different between the two readouts. And this demonstrates that the ROC-based method can be quite useful for assessing the performance of different molecules and determining the optimal threshold of activation between different readouts. This graph shows that the different readouts can provide different thresholds, but this is dependent upon the therapeutic being tested, particularly with CLN-978, which shows good activity at low receptor levels. This would suggest that CLN-978 is better able to overcome antigen escape, and we suggest that the BiTE would be more successful in treating patients with lower CD19 expression and should reduce the risk of CD19 related relapse. So now that we've discussed the individual components of the in vitro assays, I want to discuss the future of these assays and the desire to examine aspects of the TME as a whole. As the immuno-oncology field grows, there's an increased demand for more physiologically relevant assays that take into account multiple components of the tumour microenvironment rather than just the individual cells. These can take the form of co-cultures, explants, organs on a chip, or organoids, and allow for a better understanding of how the more complex systems, as well as the 3D environment, can be impacted by immunotherapies. In antibody analytics, we're taking a few approaches. One of these is the development of co-culture systems using patient biopsy material. In Glasgow, the Queen Elizabeth University Hospital is one of the largest super hospitals in Europe and has an on-site human tissue biorepository. This has allowed us to access to patient material, and as this is an NHS facility, we can also gain information on patient history that may otherwise be difficult to access through other sources. Another approach we have taken is through the development of a collaboration with a local company called Screen3D, who developed a 3D miniaturized microfluidic technology. I first want to discuss our work to develop biopsy-derived culture systems. This is still very much a work in progress, but we're aiming to develop assays with these materials that can be used to test therapeutics. So far, we've been able to successfully phenotype tissues from several cancers, including breast, colon, and pancreatic cancers, which we can process into single-cell suspensions. The benefits of this system are that we can generate disease-relevant tumour-infiltrating lymphocytes, which are a truly physiologically relevant tumour microenvironment. We were also able to cryopreserve this tissue, which opens up applications downstream. Here's just an example of the kind of flow data we're able to show from tumour biopsies. This sample comes from an adenocarcinoma of the colon, and as you can see, we're able to assess the immune cell composition and identify B cells, NK cells, as well as T cells and subsets. As I've said, we're still developing the system, but our hope is to be able to establish assays using this material shortly. As I mentioned, our collaborator Screen3D have developed a proprietary upscale lab-on-a-chip technology and associated analysis software. This technology recapitulates the 3D nature of the tumour microenvironment and can be used to create organoids, tumoroids, or spheroids. The benefits of this technology are the increased sample efficiency and scalability, which provides more information with less material. This is particularly advantageous when there is limited access to cells or tissue. The system can also examine certain aspects of drug delivery, such as perfusion, which more closely represents the conditions you would observe in vivo. Kind of models available include spheroid co-cultures, organoids, or biopsy microexplants, which can be used to provide repeated measurements and monitoring over a long period of time. Other options include sequential seeding, where different cells are added sequentially to the system to allow the creation of a 3D triculture. And these can be used not only in immuno-oncology models, but also models of other systems, including neurospheres and liver models. To give an example of the kind of applications that can be used in this system, I've included a case study focused on screening HER2 targeting low and high affinity CAR T cells against HER2 positive or negative spheroids. In this system, the HER2 positive or negative cells were seeded in microplates, and once spheroids had formed 5 days later, they were treated with CAR T cells and imaged continuously for 2 days. As you can see, the CAR T cells had little effect upon the HER2 negative cells, with cell growth remaining the same to the control, and apoptosis being only approximately 5% higher. CAR T cells also had a limited effect upon the spheroid structure. In contrast, the CAR T treatment significantly reduced the HER2 positive cells cell growth compared to the control, and had a significant impact upon apoptosis at both a low and high affinity. In terms of cellular interactions, the CAR T cells were also shown to disrupt the HER2 positive cells, indicating their impact upon the overall 3D structure. This is just one example of this application, but as you can imagine, there are many other systems in which 3D models can be employed. So that brings us to the end of the talk. I hope that I've shown you that human-based in vitro assays can be used to effectively evaluate immune-targeted cancer therapeutics. Assay design is key to their success and should be focused squarely upon the questions being asked. There's not a one-size-fits-all approach to immuno-oncology, and this should always be remembered, particularly in the selection of the immune cells and target cells in your assay. And lastly, it's worth remembering that cancer and autoimmunity are two sides of the same coin, and assays used for immuno-oncology can equally be used to assess autoimmune conditions such as RA, MS, and SLE. Similarly, a lot of these therapeutics could potentially be used in autoimmunity, and I think it's really important to always think of the bigger picture when you're developing an immunotherapy and how it can have different applications. With that, I'd like to thank Innovate UK, who were instrumental in the development of our Intex 2 system, Cullen and Therapeutics for allowing us to show their CLN 978 data, NHS Scotland for their access to human tissue via their biorepository, and our collaborator Screening3D for allowing us to share their data. I'm happy to take any questions.
Great, thank you very much for that interesting seminar, Gavin. Now it's time for the Q&A part of the session. Just a reminder to the audience, you do still have time to submit your questions for Gavin. And so to kick us off, the first question we've got here is, with Index 2, can different cell backgrounds be used other than CFOs?
Yes, so CHOs we have tended to use, like I said, simply because they don't have human antigens, so it's somewhat of a blank canvas. But we absolutely could use other cell lines, and we often use tumor-based cell lines for clients based upon their needs and what they look at. We would recommend, though, with these systems that we first knock out the endogenous receptors if that is expressed in that cell line, because you don't want interference from that cell, that receptor on your system, particularly when you're trying to look at a threshold of activation, where if you had endogenous expression, you may interfere with the threshold of activation.
Great, thanks a lot for that one. And what human materials are you readily able to access?
So materials wise, we can obviously get things from patients undergoing routine procedures. So this tends to be biopsies. In certain circumstances, these biopsies may be relatively small, particularly if they're a punch biopsy. We have access to more things when it is from, for example, a resection or a mastectomy, where you may get an entire tumour. So certain cancers like breast cancer and colorectal cancer, we also have more readily access to. We can access other cancers, but we find ovarian cancer, for example, is harder to come by, but we do have the ability to get it. And the flip side of this, in terms of autoimmunity, sometimes it is hard to get autoimmune tissues simply because of the nature of the conditions. We tend to treat patients so these tissues aren't removed. Certain circumstances where we do would maybe be Crohn's if someone's having a resection and part of their bowel removed, so we can access that.
Great, thanks a lot for that one. And in your opinion, are in vitro assays a replacement for animal studies?
So in short, no. as I mentioned, in vitro, in vivo, and in silico are three systems that are really essential to pretty much all biological research. I think in vitro systems certainly have their benefits. And I think as we move towards things like organoids or 3D culture systems, we can certainly reduce the needs for animal models. But ultimately, I think they will always have a place, at least in the foreseeable future, where we need them to answer questions about larger physiological systems and enclosed systems. I would say that, sorry, I would say that with the increase in these new systems, with the increase in AI, I think we can answer these questions in more sophisticated ways without using animals. And I would potentially say within 20 to 30 years, we could move to an environment where they're needed much less. But as it stands, I think we're going to have to continue to use them.
Great, thank you very much for that insight there. And I think we have time for just one more question, and that is, what are the benefits of the Index 2 system over traditional methods that use cell lines with different protein expression levels?
So in terms of the cell lines, I mean, like I said, we can use them, and we do use these for customers as well. I think there's several factors at play. So cell lines to access are costly. A cell license costs, on average, I think it's about $2,500. And keep in mind, if you do not know the expression level of your receptor in these cells, you may have to buy in 10, 15, 20 different cell lines to assess receptor expression, which may be zero or maybe very low. And this can cause issues, it's quite costly. And that's just the cell lines alone, that's not actually carrying out the work. There's also the issues of, if you're basing it upon literature, particularly academic literature, where you're looking at receptor expression levels, you have circumstances in which you're assuming that those cells are like the cells you would buy from a company. These cells may have been in someone's liquid nitrogen tank for 20 years, and expression levels may not be representative of that cell line in general. But there's also the titratability. If you want to look at a threshold of activation, you really can't do that with cell lines like that. But similarly as well, you will have that confounding biology in the background. The Leo paper that I had talked about earlier really had shown that when cells had a different background, that it really did cause issues where the underlying biology, regardless of receptor level, was interfering with the ability of that drug to have its effects, which in biology is fine, but when you're trying to hone down onto receptor expression levels, it really does make a difference.
Great, thank you very much. I'm afraid I'm going to have to wrap it up there, but thank you again to Dr. Gavin Meehan for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I do hope that you have found this a worthwhile session. And if you have any other questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. And remember, you can also download a certificate of attendance in the Related Resources tab to the left of your screen. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. So goodbye, and thank you once again for joining us.